Dr. Jay. You can please go ahead with your talk, sir.
Yeah, good evening, everybody, and thank you for the opportunity. Please let me know once you can see the slides. I will share them shortly. Um, the I think it's asking me to asking you guys to let me share my desktop.
You can just share it, sir. You have been allowed.
Yeah. So my background is as a biomedical engineer. I did my undergraduate education in College of Engineering Pune and graduate studies in biomedical engineering. Can you guys see the slides? Can
Not
you
yet,
guys see
sir.
the slides?
No, sir. You will have to share your screen. Share
Yeah, I
the
thought I did.
presentation.
But let me just. How about now?
Yes, we can see it now.
Okay. So really, my talk is about. I've been very fortunate to work in both coronary drug eluting and peripheral drug eluting technologies for the last 20-25 years. And so my talk is really about two topics: a drug eluting stent, Boston Scientific's Eluvia drug eluting stent, which is truly the only drug eluting stent that's available for the treatment of peripheral artery disease. And the Ranger drug-coated balloon, as you guys are very familiar with, this is one of many drug-coated balloons that's available, at least in the United States. So I'll start with the Luvia. I'm going to touch primarily on the coating aspects, but I'll touch also on the clinical implications, which at the end of the day is what's most important for you and your patients. So we start with Illuvia, and the stent platform is like any self-expanding stent. Uh, this shares the same geometry with the bare metal version of Illuvia, which is Innova. It's a laser cut nitinol stent with open cells in the majority of the body of the stent and closed cell design from at the ends. And that's the goal of that is to really optimize the forces that the stent imparts on the vessel, while making sure that it has adequate uh, flexibility for the tortuous anatomy of the SFA, and at the same time fracture resistance. The fourth thing that we designed the stent was for uniform geometry, so that you have uniform drug distribution across the length and the circumference of the treated vessel. The delivery system is a typical self-expanding triaxial delivery system, 6 French across the entire matrix, O35 guide wire compatible. What's unique about Illuvia's design is the coating that goes on there. And the coating is a polymer-based coating. Now, for those that are familiar or have used coronary stents, this shares the same exact polymer as the Promus coronary drug-eluting stent that's manufactured by Boston Scientific, as well as the Xience coronary drug-eluting stent, which is manufactured by a competitor, Abbott. And the unique properties of this polymer are that it's extreme vascular compatibility. It's known for its hemocompatibility, and it really, at this point, more than 20 million patients have had either a Promus or a Xience stent. So there's a long history of good biocompatibility. One of the key features of having a polymer on this stent is that it allows you to do a couple of things. One, it allows you to have a very low dose density of the drug. So if you look at the measured dose density of the stent, it's about 0.167 micrograms per millimeter square of the stent surface area, which is really, really low. The other thing it does is it allows a very localized delivery of drugs. So, for example, if you look at the mechanism of how the drug is delivered, it is through a diffusion gradient. You have high concentration of drug in the polymer and none in the tissue. And so as the polymer gets hydrated upon implantation, it will swell and then it will allow that drug to be distributed from the stent. So it's a very controlled mechanism. Over time, of course, that drug dose will completely leave, so you can only control it for a certain period of time. And so the question for us in terms of a challenge is, how long do we allow this drug to, how much drug do we deliver? In this case, paclitaxel, and for how long do we do it? And so, just as a frame of reference, on the coronary side, both with Promus or Zion stents, that delivery time frame is about 3 to 6 months. And so the question for us was, does it make sense to have that same regimen that we do in the coronaries? But as we have learned from physicians like yourselves, the SFA is a completely different pathophysiology compared to coronary artery disease. And one of the things we learned as we were doing the research is that the timing of restenosis in stents, especially in peripheral stents, seems to be a little bit longer. So we found this research done by a professor in Japan, Professor Ida, and what he found out in his studies was that if you look at when stents are most likely to restenose in the SFA, it can happen early or late, but the maximum probability tends to be around that 1 year time frame. I'm sure this is reflected in your practice, and so this is one of the drivers for us to figure out how to dose it. The next curve I'm going to show you on the right is a PK curve. And a PK curve measures the amount of drug that's being delivered over time. So on the y-axis, you have the actual measured paclitaxel, and obviously this is done in a preclinical study in a pig typically, since we can do that in human. And then on the x-axis, you have over time. And what I'm showing you in the gray is a typical DCB curve. It doesn't really matter what company does this. This is typically a high bolus of drug, and then most of that drug is delivered in that 60, at the most 90 days. If you look at Zilver PTX's own data, this is Cook's drug-coated stent, what you see is the similar elution profile. You have most of the drug, and most of that drug is delivered by 60 days. This is well published. Cook has published this over many years. And you can see most of that drug will therefore be gone in about 60 days. Well, our understanding is that the disease happens much later in the SFA. And so when we designed Illuvia, the light blue curve is how we design the elution profile. So we provide a bolus in the early stages because that's when injury occurs upon implantation of a stent or atherectomy or balloon angioplasty. But then we sustain that release of drug for approximately 1 year. Now that can vary based on patient characteristic and lesion characteristics. But this is the differentiator of Illuvia, and we believe this is really the secret sauce of what has allowed us to have really outstanding clinical outcomes. And again, I'll go into a little bit more data on how specific this delivery of Illuvia is. And one of the ways that I think you can see this is what you're seeing on the left is the same data I showed in the previous slide. The green bars at the bottom represent the stented zones only. The purple and the red curves bars are 5 millimeters outside the stent. So what you can see is most of the delivery across the time frame of the drug delivery occurs in that stented zone alone. Very little, there's obviously some diffusion of the drug outside of the treated lesion, but it's only less than 5, it's primarily in that stented region. Most of the curves are bars you can see are green. So this is the tissue PK. And so again, just highlights that the beauty of having a polymer-based delivery system, where most of the drug is delivered in the stented zone, where you want the drug to be not elsewhere else, not anywhere else. The other PK study that the FDA actually requires us to do is a blood PK. What I've shown you on the left is tissue PK, actual measured paclitaxel levels in a pig model. What's on the right is how paclitaxel actually leaves your body. and it's a drug that's really fairly easily metabolized. One of the beauties of Eluvia is that 30 minutes after implantation, you cannot measure any paclitaxel in blood. With drug-coated balloon, it's a little bit slower. So typical drug-coated balloons, for example, Ranger will take about an hour before you do not detect paclitaxel in blood. With Impact, it's 2 to 3 days. Same thing with Silver PTX. Now, this is not to suggest that there is any harm in doing that, but what it does highlight is that this is not a very efficient way of delivering drug if a lot of your drug is still in your circulation 2 to 3 days after implantation, rather than in the tissue where we really need it. So again, highlights again the really the beauty of the polymer in being able to provide a very metered, localized delivery of drug to your lesion where you really want the drug to be. A comparison, now this is meant more as a comparison of the different technologies. On the first column, you see Eluvia, which is a polymer-based delivery system. And as I had highlighted before, the drug dose density is very low, it's 0.167. Silver PTX, because it does not have, in the second column, it does not have a carrier or polymer or an excipient, has a very high drug dose, relatively about 18 times more, 3 micrograms per millimeter square. You can see that the downstream particulates therefore tend to be a little bit higher with Silver PTX. Again, not to suggest that does any harm, other than to highlight that it's just a less efficient way of delivering the drug. And then on the third column, I'm showing you a typical DCB, maybe one with the highest drug dose. Impact, as you guys are aware, is 3.5 micrograms per millimeter square of the surface of the balloon. And again, the key thing I would highlight is if you look at how long the drug stays in the tissue with each of these technologies, it's 12 to 15 months for Eluvia. It's only less than 60 days for Silver PTX, and it's about 60 to 90 days. So really, this is, I will go into a little bit more detail on drug-coated balloon technology next, but I think this is a nice overview of the differences in technology. Eluvia is truly the only drug-eluting stent because it elutes for a period of time, just similar to how coronary stents do it. Silver PTX was a pioneer when they first came out, but it's clearly a drug-coated stent, not a drug-eluting stent. And of course, all DCBs are, by their very definition, coated technologies. They do not enable a sustained release of the paclitaxel in tissue. I think this is really more about the technology, but I want to touch on the key question that many of you may be asking. That's great that there are differences in technology, but what do the clinical outcomes look like? And others may have spoken to this, but really one of the first head-to-head clinical trials was done with Eluvia against Silver PTX. This was done now several years ago. It was a randomized controlled trial globally done, about 500 patients that for the first time compared Eluvia with Silver PTX, the only other paclitaxel stent that was available, that is still available in the United States. And even though this was not designed to be a superiority study, it was really designed to be a non-inferiority study. The post hoc analysis found out that Eluvia demonstrated a statistical significant primary patency compared to Eluvia. And I think that's the proof of the pudding. I can talk about the design and how unique and targeted the delivery of the drug is. And as I will highlight in the next couple of slides, it's really the clinical data that shows how outstanding the outcomes are. One of the things that I also wanted to highlight with the clinical trial data, this is the imperial data, and now 5-year data is available, is how consistent the outcomes are, irrespective of the lesion type. As the lesions get more complex, the outcomes don't really change. So, for example, the long lesion subset is again 90% plus primary patency. Same thing with diabetics and chronic total occlusions. CTO is probably one of the more challenging subsets, is probably at the low level. But other than that, I think consistently, and I always call this coronary-like outcomes, 90% plus primary patency, TLR rates in the single digits for the most part. Um, a couple of other slides that I want to, and we don't have enough time to go through all of these, but all of these are available in the public domain, published or presented at different conferences. I think historically what we have seen as on the x-axis, if you look at the complexity of both lesions and patients, as that complexity increases, the outcomes, typically with bare metal stents or even with silver, continues to go down. And what I think we take special pride in seeing is that, irrespective of lesion complexity, the the outcomes continue to be consistently 90% plus primary patency. Even the TLR rates, when we look at them at 2 years, are really in 90 plus or less than 10%. So really, most of these patients are not coming back. Even at 2 years. So really outstanding outcomes in a very complex patient and lesion population. Um, this just highlights the body of work. I think we invest fairly heavily in technologies, but as much or more we invest in generating the clinical data to convince you that this is truly outstanding technology in terms of delivering truly truly great outcomes for your patients. And again, there's a number of studies, some done by Boston Scientific. You guys are familiar with this pyramid that shows the level of data. Both from randomized controlled trials as well as non-randomized registries that has been generated. Again, I'll switch over to Ranger, but I'll pause for a second to see if there are any questions that I can answer, and we can always come back to Illuvia if 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 there are any questions. Yeah, what I'll do is I'll go very quickly through the range. The history of this is drug-coated balloons are the concept of delivering antistenotic therapy is not new. Boston Scientific has been working on it since the mid '90s. These are published papers, a couple of different versions, and none of these really worked. And the challenge that these didn't really work is because your endothelial layer tends to be a pretty big barrier to delivery of drug. And this is where I say, unlike Illuvia, which is a drug-eluting stent based on a polymer, the mechanism here is that you coat the balloon with a crystalline form of paclitaxel. The excipient is like a binder, right? Just like aspirin is not 100% salicylic acid, it has things like cellulose to allow delayed delivery of drug. All these binders are doing is making sure that the drug is not lost during transit. I thought there was a question on how much is lost. A significant amount of drug is lost in transit, both upon entering the sheath as well as upon inflation. But you still get some drug there, and the mechanism is that you get crystalline drug embedded into the vessel wall. So that's the mechanism. The image on the right on the bottom shows that the green image is actually crystalline paclitaxel, and over time that will dissolve. This is where the excipient is important. If you don't use excipient, again, this is a PK curve similar to what I showed you before. The green and the black are ones that didn't have an excipient, and you can see you get very little tissue retention, very little in the tissue. As soon as you add an excipient, you can extend that duration of release. And so that's the criticality of the excipient. Every company uses a different excipient based on their patents and intellectual property. We use something called ATBC, and that's really allowed us to do two things. One, maximize how much we get into the vessel wall and minimize how much goes downstream. But it is going to be, I will tell you, even though work on Illuvia and Ranger, both Ranger DCBs in general are not as efficient a way of delivering drug as Illuvia or a polymer-based system is. This is upon hydration. Obviously, this addresses your question. What happens once because the coating will come in contact with blood or fluid in this case, as soon as you hydrate, again, you see that some of that coating is going to come off. At the bottom, you see flaking from the impact coating, which uses urea as an excipient. And then if you look at downstream particulates, this is what's lost either during insertion into the sheath or upon inflation. And I will transparently tell you, this is well published. A majority of the drug is with all drug coated balloons, as well as with silver drug coated stent, is lost downstream. It's less for Ranger, even though the drug dose is the same, and that's based on the choice of excipient. So, for example, Lutonix has a very water soluble excipient, and so it tends to lose more of the drug, even though these guys have the same exact drug dosing. You lose a lot more with it. Lutonix. Lutonix is the only drug-coated balloon that requires you to get it to the lesion within, I forget whether it's 120 seconds or 180 seconds. We don't have that restriction with Ranger or Impact or any of the other DCBs, only with Lutonix. And so again, that is important from a safety perspective, perhaps. What is more important is how much drug do you deliver into the vessel wall? And this is where, even though Ranger has half the dose on the balloon surface as Impact, you can see that most of that, there's no difference in terms of the PK curve. It's the same amount of drug is being delivered by both these devices actually into the lesion. And so this is, again, preclinical or pig data. I won't spend too much time on the delivery profile, but what you can see is the Ranger has the highest primary patency now out to 5 years. And this is, I'm doing a little bit of an apples to orange comparison. This is every company's clinical trial that was submitted to the FDA. So again, highest primary patency with Ranger compared to all the other devices. I know this, again, I'll pause. I know you guys are running late. What I'll say is, like Illuvia, we've built a strong base of evidence on the use of both these technologies for the treatment of peripheral artery disease. And I'll stop there, but I'm happy to answer any questions that you might have.